Hello everyone. If you're a first year medical student, you know how confusing it can get to identify histology stains during practical exams. In this video, I will break down 7 of the most important stains you must know and show you exactly how to identify each one in a slide. I will keep it super simple, just the name, key color clues, what the stain highlights and how to tell it apart from others. So let's make stain spotting easy and exam ready. So let us start. Our first stain for today is your Hematoxylin and Eosin dye stain. So Hematoxylin is a basic dye, Eosin is an acidic dye. So Hematoxylin binds to negatively charged structures like your DNA and RNA and it stains the nucleus blue-purple. As we can see over here, the nucleus is stained as blue and purple. Eosin binds to your positively charged proteins in cytoplasm and it stains the cytoplasm pink as we can see in this image. What is the stain used for? It is the most routine stain used in histology for examining overall tissue structure and your cellular morphology. Key identification feature is your nucleus is blue to purple and your cytoplasm is pink in color. This is a very easy stain. Next stain is your Van Gieson stain. So it uses acid fustian and picric acid. Acid fustian stains the collagen red as we can see over here in the image, while picric acid stains the muscle and cytoplasm yellow. We can appreciate it over here. What is it used for? It is used to differentiate collagen fibers from muscle fibers in your connective tissue sections. Key identification features is your collagen is red in color and your cytoplasm is yellow in color. How to differentiate it from other stains? Unlike Masson Trichrome stain which uses 3 colors, Van Gieson uses only 2 contrasting colors, making it simpler and a quicker method. Our next stain. The next stain is your Mason's Trichrome stain. It got its name Trichrome. Why? Because it uses 3 dyes that have been used. Mechanism of action is it uses a combination of acid dyes to differentiate stains tissues. Aniline blue or light green binds to the collagen. Acid fustian stains muscle and a nucleus stain like the Viga's iron hematoxylin it stains the nucleus. What is it used for? It is used to differentiate collagen, muscle and epithelial tissues with high clarity such as in liver fibrosis or tumor stroma evaluation. Key identification feature is your collagen is blue or green in color, muscle and cytoplasm stains red and your nuclei is black in color. As we can see in this image, this is your muscle and the cytoplasm that is stained red. Nucleus is stained black and collagen fibers are stained blue or green as we can see over here. Next is your Mallory stain. This stain looks very similar to that of your Mason's Trichrome stain, but there is a differentiating feature, that is the dye used in the Mallory stain and in your Mason Trichrome stain. Let us understand how. So your Mallory stain was named after Frank Burr Mallory. Mechanism of action, it uses a combination of acid fustian, aniline blue and orange G to differentiate stained collagen, cytoplasm and red blood cells. What is it used for? It is used to identify collagen fibers, fibrosis and connective tissue changes in organs like liver and skin. Key identification point is collagen stains blue in color, cytoplasm stains red in color and RBCs are orange in color. Why is it used? It is a strong stain that is used to differentiate the contrast between collagen and other tissues, valuable in detecting fibrosis and scarring. Now over here is how to understand and differentiate between Mason Trichrome stain and your Mallory stain. Both are trichrome stains, as it is in the name it suggests, both of them are your trichrome stains. But Mallory stain collagens deeper into deeper blue and RBCs are orange in color, while Mason Trichrome stains collagen blue to green and the muscles are red in color. Mallory gives a more vivid blue for collagen and is especially preferred in liver and kidney pathology. So Mallory stain you have to remember 2, liver and a kidney pathology. Now over here is a table to differentiate between both the stains. So the key differentiating factor, one is the dye that is used. The second one is your muscle that is skeletal and smooth. In your Mallory stain, muscles are usually not seen distinctly, while they are distinctly seen in your Masson's Trichrome stain that is red in color. The special features are the Mallory stain highlights the connective tissue cells and the Masson Trichrome is evaluating fibrosis. Kindly go through this table carefully. That will help you differentiate between your Mallory and your Masson Trichrome. And another key feature, like when you're said in your exams to identify this which stain, usually in your Mallory stain you will see RBCs are seen that are orange in color. But as we can see in your Masson's Trichrome stain, the orange or the red colors of RBC is not well appreciated. Okay, the next stain is your poor iodic acid and Schiff stain. This stain is used for what? It is used to detect glycogen in your mucin, fungal cell walls and in your basement membrane. The mechanism of action is poor iodic acid. It oxidizes 1,2 glycol groups in carbohydrates to aldehydes. Schiff reagent reacts with aldehydes to produce a magenta color. So the slide of this stain, you will usually see it is magenta or pink in color, while that of your hematoxylin will have both pink and purple in color. But majority of your PAS stain is what we can appreciate over here is magenta or pink in color, as it represents the carbohydrates that is present. In this image, we can see the gastric slide, esophageal candidiasis slide and the glycogen in your liver. Key identification point is the structures containing carbohydrates appear bright pink or magenta in color. How do we differentiate it from other stains? PAS stain is specifically used for carbohydrate-rich structures which remain unstained or faint colored in your other routine stains. Your next stain is your von Kossa stain. So it is named after Julius von Kossa, who developed this method to detect mineralization in your tissue. Von Kossa, very easy. You will see the calcium deposits that are black in color. Mechanism of action is silver nitrate. It reacts with phosphate or carbonate ions in your calcium salts, which are reduced to black metallic silver under light exposure. So silver nitrates will react with your phosphate or carbonate in the calcium salts, that will be reduced to your black metallic silver color under your light exposure. It detects anions, not the calcium ions. What is it used for? To identify the calcium deposits in your bone pathology, dystrophic calcifications, atherosclerosis and in some tumors. Key identification point is your calcium salts appears black while the background tissues remains light in color. Now, how do we differentiate it from an alizarin red S? That is also used to detect the calcium deposits in your tissue. So, your alizarin red stain will have directly red color of your calcium deposits, while your von Kossa stain will have black color for your calcium deposits. Thank you guys.